Hey everyone, in this video I am going to talk about an important technique in molecular biology, and it is called Southern Blotting. So Southern Blotting, this technique was developed by a scientist named Edward Southern, and the technique is named after him. So essentially this technique is used to detect, a specific DNA molecule among a population of DNA molecules. Now, for example, let's say we have this imaginary sequence called Super Awesome Sequence 1. Super Awesome Sequence 1. And maybe it's not a gene, maybe it's some sequence in the human genome, and we don't really know what it does, and not all humans have it. So let's say it's known to be about, 2,000 base pairs long. And it's found in some humans, but not others. Well, we could perform, or we could use Southern blotting to see, which humans have Super Awesome Sequence 1 and which do not. So essentially, how we would start, well, for example, let's see what would happen if we took DNA from 3 humans. Let's say human DNA, we digest it with a restriction enzyme. And we want to do that, right? Because we want the DNA to be in relatively short fragments for this assay. So let's say we have a gel here with 4 lanes, and we'll put our DNA ladder in the leftmost lane. And let's say we get DNA from 3 volunteers. Let's take me and 2 of my friends from 2 of my childhood friends, so Jim and Adam. Okay, so we got DNA from these 3 people, me and 2 of my high school friends, and we digest the DNA with a restriction enzyme. It doesn't really matter what enzyme we start with, but let's say EcoR1. That's one we've seen in the past. Now, when we visualize the DNA on the gel after performing gel electrophoresis on the DNA, and remember, with gel electrophoresis, we would load the DNA into a well, and we would apply a current, and the gel electrophoresis would separate the DNA molecules by length. So we might see something like that. We could use a dye like ethidium bromide, which binds DNA, and it would light up all the DNA molecules. And these little squiggly lines I'm drawing, well, this is what the gel would look like. And why does it look like this? Right? Well, essentially the EcoR1 enzyme is cutting like all over the genome wherever there's an EcoR1 recognition site, and so we're getting DNA of essentially many different lengths, right? And how can we tell if SAS1 is somewhere in here, right? Well, we can't, right? We just see all the DNA. In order to determine if SAS1 is in here, well, we would need to perform a Southern blot. Okay, so how does that work? So essentially what we're going to do is perform these 2 steps like I just went over, but then we're going to need to transfer the DNA to a solid membrane, like a nylon membrane or a nitrocellulose membrane. And then we're going to use a probe that is specific for the SAS1 sequence. And it's going to cause that SAS1 sequence essentially to light up if it's present. And there are a couple different types of probes. There are chemiluminescent probes, which are more common nowadays, and there are radio-labeled probes, which will allow us to detect the SAS1 sequence by radioactive decay. And so the technique we're going to go over is the radio-labeled probes. So, okay, let's go over the various steps of the Southern blot. So, like I said, we need to digest the DNA with a restriction enzyme. We need to separate the DNA by gel electrophoresis. So we're going to have, at this point, we're going to have what I just showed you, right? We're going to have something like this. Okay, so now what we need to do is soak the gel in a sodium hydroxide solution. And that's pretty easy, right? So we can pick up that gel very gently. We don't want it to break. And we can put the gel in some sort of tray, often a glass baking dish, like a 9x13 baking dish can be used, or a 9x9 baking dish that you might use to, bake brownies or something like that. Obviously, you'd have one for the lab. You wouldn't bake brownies in it, but you'd use it for Southern blots. So let's assume this is the gel tray, and we have or the glass baking dish. We have it filled with a sodium hydroxide solution, and we just pick up the gel, and we can gently lay it into the solution. And the sodium hydroxide, what that is going to do is it is going to denature the double-stranded DNA molecules in there. So the hydrogen bonds that hold the two strands of DNA together, well, they can't form in the sodium hydroxide solution. So the sodium hydroxide solution will break those, or they can't remain formed in the sodium hydroxide solution. So the sodium hydroxide solution will break those bonds, and the strands will tend to separate from each other. They won't go very far, but they won't remain next to each other, so they could easily form hydrogen bonds again, or by complementary base pairing, when the sodium hydroxide solution is removed. So, long story short, sodium hydroxide solution separates the double-stranded DNA into single-stranded DNAs, and that's going to be important for detecting the target molecule, with a probe. Okay, so after that step, what we need to do is essentially neutralize the sodium hydroxide. Essentially what we're going to do, we're going to take the gel out, and we're going to submerge it in a solution of transfer buffer. And the transfer buffer is just a high salt solution. And the high salt inhibits DNases, things that might degrade the DNA, and also allows for the DNA to stick to the membrane that we're going to use as part of this process. So after the gel has been neutralized, we need to transfer the DNA to a nylon or nitrocellulose membrane. So let's just we're going to talk about nylon. They essentially work the same way. Now, these membranes, essentially they're sheets of paper. They're like sheets of paper. They're about as thin as a sheet of paper, but they're made of nylon or nitrocellulose instead of cellulose. And when they're wet, they don't really tear. They don't tear very easy. So you really need to cut them with scissors if you want to tear it. So how does the transfer process work? Okay, So it's really neat. So I used to do this a lot when I was a PhD student. So essentially, I might need this sheet of paper in a second. So essentially what we need to do is use some sort of dish. I like to use a glass baking dish for this also. And so I'm going to diagram the glass baking dish like that. We need to have a tray of some sort, and this tray will have little holes in it, so you can put the tray upside down. It's going to sit in this buffer, the transfer buffer. Transfer buffer. And we have little holes, so if there's any air trapped under this tray right here that's been inverted in the buffer, the air can escape and this tray won't float. So, and then we're going to get this blotting, we're going to use blotting paper. Now blotting paper is a sort of a thick paper that really absorbs liquid really well. And so this blotting paper, this thick paper, we're going to lay it over the tray, and we're going to hang the ends of the blotting paper over the tray into the water here, into the transfer buffer. And so this is going to start sucking up the buffer by capillary action. Now on top of the blotting paper, we are going to have our gel, and let me color the gel blue. So that is our gel, where the DNA has been denatured with sodium hydroxide, and then the gel has been neutralized, so the DNA is all single-stranded in here. And on top of the gel, we're going to put our membrane. And the membrane, in this case, it's going to be a nylon membrane, and it's pretty thick right here, but it's about as thin as a sheet of paper. It's cut to the same size as the gel. On top, and okay, let me label that nylon membrane. On top of the nylon membrane, we are going to have a sheet of blotting paper. So the same paper here is going to be on top. I will color that the same as the blotting paper is usually white, but I'll color it gray here. Now that is cut to the same size as the nylon membrane, and on top of the nylon membrane, we're going to have a lot of paper towels, just regular old paper towels. You know, if you go to the restroom, there are usually paper towels there. Yeah, just that. And they're all dry, so it's important that they're dry paper at this point. And so a big stack of paper towels here. Now everything is cut to the same size. The towels are, I didn't diagram it that way. I should have done a better job, but everything is cut to exactly the same size. And I'll tell you why in a moment. On top of the paper towels, we put some sort of book, a textbook, or some sort of weight that can apply some force down on the paper towels. Not a lot, just enough to keep everything in contact with each other. So all the paper towels and the blotting paper and the gel and the nylon membrane. And I should say, as we were putting this together, we would sort of squeeze out any air bubbles that may have formed. Now the transfer buffer, now all of this, the paper towels are dry, and the transfer buffer was soaking up the, the blotting paper is soaking up the transfer buffer by capillary action, and the transfer buffer is going to flow up into the paper towels, and it's going to carry the DNA molecules along with it. But the nylon membrane is positively charged. The DNA molecules are negatively charged, right, because of the phosphate backbone. So the DNA molecules are going to stick to the nylon membrane. And then this takes a couple hours. Usually it's done overnight. And so the DNA will slowly move up with the transfer buffer into the paper towels, but the DNA will stick on the nylon membrane. And can you think, why does everything have to be cut to the same size? Well, for example, what if these paper towels here touched the blotting paper down here? What would happen is the buffer wouldn't go through the gel and the blot, or wouldn't go through them very efficiently. It would take a shortcut up to the paper towels. And that's sort of called a short circuit. And so everything is cut to the same size to force the transfer buffer up through the gel into the paper towels that way, so it can carry the DNA molecules onto the membrane. Okay, so that's how we would do the transfer. That's how the transfer is usually done. And there are other apparatuses that can use vacuums, but this is the easiest and I think most of one of the maybe not the most time efficient, but one of the easiest methods to transfer the DNA molecules from the gel over to the nylon membrane. Okay, so let's see, where's my other piece of paper that had the notes? Okay, here it is. We were at transfer DNA to nylon or nitrocellulose membrane. We're using nylon in this example. Okay, so the next step, what we need to do is we need to expose membrane to UV light. Essentially, we need to take a UV light. Usually it's called a cross-linker, which is a little microwave type thing that will add the right amount of UV light or shine the right amount of UV light onto the blot or the membrane. But essentially what we're going to do that, we need to take this apart. And so what we would do is take the filter paper, take the paper towels off, take the weight off, take the top blotting paper off, and then we're left with something that looks like this. So if we're looking down on this right here, and let's say the solid support is down here, and the blotting paper is sitting in the buffer down here. So what we need to do is just peel this up, and peel this membrane off of the gel. And so that would look something like this. So we'd be holding the corner with some forceps or something like that, and we just peel it up and peel it off, and oh, we have our membrane. We'll have our membrane. And that's going to look something like this. I'm cheating a little bit here to speed things up. What are these notes here? I don't know what those are. Okay, so here's our membrane, and I'm showing, we can't really see where the DNA is. Maybe if we wanted to use ethidium bromide, we could visualize the DNA on the membrane. And at this point, we can call it a blot. So they're nylon membranes, nitrocellulose membranes. But once the DNA is on there, we're calling that the blot, the DNA blot. Now, okay, over here we had our ladder. Here's all the DNA molecules. And what we would need to do is shine UV light onto the membrane. And the reason we do that is that adds covalent links between the DNA molecules and the nylon membrane. It covalently links the DNA molecules to the nylon membrane. Because remember, before they were sort of held together by the positive and negative charges, by the positive charges of the nylon and the negative charges of the DNA. But the UV light will covalently link the DNA to the membrane. Now, remember, the DNA molecules are single-stranded because of the sodium hydroxide treatment. And we need it to be single-stranded because now we're going to use a probe, a radio-labeled probe, to see where our target sequences are, or if our target sequences are found in the genomes of our three individuals here. Let's say Tom, Jim, and Adam DNA. Okay, so we're going to talk a little bit about how to make a DNA probe, or a probe for Southern blotting, but let's skip right to using the probe to detect our target sequences. Okay, so here is the blot. I like the term membrane better, but here is the membrane. Here is the membrane. And what we can do, and often now what we'll do is use these glass hybridization bottles. But for many years, what was used was plastic bags that you could seal with heat. So this is the plastic bag, and you put it in a little machine that presses down on the bag and it seals it, so it's leak proof. So this is sealed right here. So it's a Ziplocs you can open and close, Ziploc bag, or they have those little plastic zipper like things. Those are, you can open and close them many times. With this heat sealed bags, it's permanently sealed, and you need to cut them open with scissors. Okay, so let's say in this bag we have hybridization buffer, plus our probe. And now the probe could be double-stranded or single-stranded, but if it's double-stranded, it needs to be denatured into single strands for the hybridization process. Okay, there is our probe, and let's say the probe is specific for the SAS, the super awesome sequence 1 sequence. I don't know if I added 1 before, but we'll call it SAS 1. So let me add some colors here. I'm using this color for the membrane. I'll use blue for the transfer buffer. And then we would have to incubate this probably overnight, maybe at like 50 degrees Celsius in some sort of shaking incubator. And the probe, both strands of the probe are going to be just floating around in the hybridization buffer, and they're going to tend, whenever they come into contact with the complementary DNA, which is the SAS1 DNA, when they come into contact, they will stick, they will hybridize. That's what we usually use this term hybridize. Sometimes we'll say anneal. You can use these interchangeably, but hybridize, we'll use hybridize here. And so what that means is the probe is just going to stick to the complementary DNA that has been cross-linked to the nylon membrane. And so ideally it doesn't stick anywhere else. And the hybridization buffer is going to contain some things that keeps the probe from binding non-specifically to the membrane. So the probe should only stick to the membrane if there is SAS1 DNA on the membrane, and it would only stick to the part of the membrane where SAS1 sequences are, because it's going to hybridize to those SAS1 sequences if they are there. Okay, so after that process is over, what we will need to do is essentially wash the membrane to remove any extra probe, to remove any probe that happens to be sticking to it in a non-specific manner. And so you can do that any number of ways. You can use bags, you can use bottles, you can stick the membrane into a glass tray with the wash buffer. And this is kind of a pain, right? This is kind of a pain because, especially if you're working with radio labeled probes, because everything has to be, all the work has to be performed behind these thick plexiglass shields, and all the buffers need to be disposed of as hazardous waste. So it's kind of a pain, but it's very useful, very useful. Okay, so after we wash off the excess probe that's bound non-specifically, we can take the membrane, wrap it in saran wrap, and expose it to auto radiography film. Auto radiography film, sometimes called X-ray film. Auto radiography film. So expose it to auto radiography film. And what that type of film is, it has these sort of silver halide molecules on it. And when radioactivity decays, it converts the silver halide form into a form of metallic silver. And so anywhere on the film that we detect metallic silver, well, that's where we know radioactivity was. That's where we know radioactivity is located on the membrane. You can also hold a little detector called a Geiger counter up to the membrane, and it should, normally a Geiger counter, when there's no radioactivity around, will go like beep, beep, beep. But if we hold it up to this, well, it should be going like this, beep, 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 beep. And that's a little scary, but again, you've got these giant plexiglass shields around, and you're protecting yourself from the radioactivity when you're working with this. Now, okay, the film, we're going to expose film to the membrane, and then we're going to develop the film and take a look at what it looks like. Okay, so let's say here is our film, here is the blot, here, this is where the wells were located. We have Tom, Jim, Adam. Sometimes the latter will even show up a little bit because there's so much in it's kind of, you get a little bit of non-specific binding sometimes to the probe. But that's advanced, so we don't really need to talk too much about that. But okay, let's say what we see is a nice band around here and a nice band around here. Well, we would say, okay, looks like Jim and Adam have the super awesome sequence, SAS1 sequence in their genomes, but me, unfortunately, I don't have it. Okay, well, then we need to figure out, why do they have super awesome sequence 1 and I don't? It's not very fair. Anyways, that's how Southern blotting works. And okay, in the next video, I'm going to show you how we can use a process called DNA Nick translation, or just Nick translation to make a radio labeled probe for Southern blotting.